You're listening to the Effective Statistician podcast, a weekly show with Alexander Schacht, myself, and Benjamin Pieske, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today we will talk again about Estimands, but not in the clinical trial setting, but more in observational studies. And for this, I'm really happy to have Artemis Koukounari on the show, with whom I will talk about this. So stay tuned for this really insightful episode, even if you're not working on observational data. Understanding estimands is really important in the clinical trial space as well as in the observational space. And if you want to understand it as well as communicate it, implement it effectively, and potentially even driving change in this area, then I have some good news for you. We have the Effective Statistician Estimands course, and this course is not just by myself, no, it's together with Kaspar Rufibach, who has done a lot on this topic across the industry, as well as in his company at Roche, where he successfully implemented lots of these different things. So check out the Effective Statistician Estimand course, where you can learn a lot about this topic. By the way, the first couple of videos on it are free, so you can just sign up for these, have a look into these, and if you like it, then you can look into whether it's a good investment for you, both from money as well as from a time perspective. I'm producing this podcast in association with PSI, a community dedicated to leading and promoting the use of statistics within the healthcare industry for the benefit of patients. Join PSI today to further develop your statistical capabilities with access to the video-on-demand content library, free registration to all PSI webinars, and much more. Head over to the PSI website at psiweb.org to learn more about PSI activities and become a PSI member today. Welcome to another episode of the Effective Statistician. Today I'm really excited to talk with another listener of this podcast and someone that also has a lot of knowledge in an area that I have worked a lot in. Welcome, Artemis. How are you doing?
I'm really good. Hi, Alexander. Thank you very much for having me in your show.
Very good. And today we are talking about two topics that come together here. Real-world evidence or observational research on one hand, which is a pretty hot topic for many years, but it's only growing hotter over the last years. And the other is Estimands. I think the Estimands topic, of course, came from the clinical trial area, but it's similar important, or maybe even more important in the observational area. And what I really love about the Estimand framework is that it kind of pulls a lot of methods, approaches, problems that have been tackled in observational research for many years into the clinical trial area. And so I love that, here we are connecting the dots in a much better way. Before we go into the content part, Artemis, please introduce yourself.
Thank you. Thank you very much, Alexander. I have most of my experiences with observational research. I have studied, have a BSc and an MSc in statistics, and then I did a PhD in statistical epidemiology. And the last 3 years, I have transitioned to the pharma industry from academia. And now I work as an associate director in real-world evidence with Novartis. So it's my great pleasure to be here with you and talk about this topic, very important topic.
Awesome. So let's dive into where we're coming from. Yeah. So in terms of observational studies, what is your experience around the history of Estimand type questions we have there, both in prospective and in retrospective observational studies?
Yep. In retrospective secondary data use, I have been involved in teams looking at comparative effectiveness, also on hybrid controls where you have actually early phase trials where the sample sizes are really small and you try to enhance the existing control with historical data, either from trials or real world data. So over there again, the target of inference becomes very important. Who do you want to estimate the treatment effect for the overall population, for just the treated, and so on. In terms of prospective studies, currently I'm part of a team that tries to set up multi-country primary data collection, where again the main aim is comparative effectiveness. However, the fact that it is from multi-country sites creates a lot of issues with the inherent assumptions of propensity score methods, like, for instance, the consistency assumption, where you must have clear treatments, becomes a nightmare of having the same standard of care in all the countries involved. Yeah. The positivity assumption is under doubt because there are different access issues. So, do all the patients have the same probability of getting the treatment under question? And then, the main assumption of exchangeability and measured confounding, can we take into account the confounders that are needed in order to implement these propensity score methods to achieve balance between the treatment groups and be able to infer causal treatment effects? But in all these experiences, I feel that we are not driven by, we first define the Estimand and then we couple the methods associated with it, or what are our stakeholders want? What is the research question that they impose that we compare it with the Estimand? So I think, as in the world of trials, we really ought to become more serious about defining first the Estimand as an iterative process and then looking at what methods can back up the target of inference. And also in terms of retrospective studies, another experience that I had was trying to extrapolate the findings of a trial to a target population with real-world data. So over there, the Estimand was the targeted average treatment effect. And there are issues. How are the covariates that you are looking from the trial to the real world measured? Are they measured in the same way? Do you miss some? Are the effects the same? So
there's a lot of other topics. Yeah, I am just thinking back in terms of my early time in observational research, where we also did comparative observational studies. And I'm just thinking about a particular one that I worked on about 20 years ago, really big schizophrenia observational study with many different treatments. And at the start, it was pretty easy. We looked into 3 months follow-up data, and in the first 3 months after starting therapy, nothing changed a lot. Yeah, so it was pretty easy. We look into the data as based on the treatment policy, more or less. What you start with, we analyzed it accordingly. Yeah, but then, of course, the first problem was, okay, in an observational setting, you don't have a randomization. And so you do the propensity scoring approach to compare apples to apples, similar patients. And thinking back, we had then these different propensities categories, having the reference treatment probability of 0 to 20%, 20 to 30%, and so on. Thinking back, we just pooled across these, That's it. But we never really thought about, okay, does pooling actually make sense? Yeah. Is there treatment by propensity score interaction? And if there is, well, that is pretty interesting, yeah, because then, of course, the treatment effect depends on your propensity score. Now, the propensity score is not something that, you can give to patients easily. Yeah, so you can't say, well, can't look just into their data and say, hey, you have a propensity score of that, and therefore your treatment effect will be this. It's a calculated score. Yeah. So that comes with completely different topics than with the typical things in clinical trials. Yeah. Where you have randomized comparisons. Unless, of course, you also do propensity scoring there. You get into the same problem. Yeah. The other thing that I found really interesting that I learned from observational studies was this kind of survival bias. Yeah. So
Yes.
in the end, this observational study expanded not just for 3 months, but for 3 years. And already after 1 year, if we compared the patients that started on a medication and that were still on the same medication at 1 year, they were all the same. Yeah.
Yes.
There was no treatment effect whatsoever anymore.
Yes.
And then the physician said, well, that's pretty clear because if you haven't responded, yeah, and you're not working well, then you will switch. So more or less, because of the standard of care of the practice, by design, you don't see any differences. That's the kind of on treatment Estimand after 1 year. And this kind of thing was pretty useless because everybody would be responding.
Yes.
And so, yeah, that showed that, this kind of problem about different Estimands. And yeah, only more than 15 years later, this kind of thing emerged around the Estimands. So how do you see it? You mentioned one thing that is the positivity assumption. Can you speak a little bit more to what this is and why that might be a challenge?
Well, the propensity score methods have, nothing comes for free. It's a very nice method of trying to tackle confounding and measured confounding. Whatever you measure and achieve balance of those confounders among the treatment arms, but it comes with assumptions. The main assumption is, one of the main assumptions is the exchangeability, measured confounding and conditional dependence of these covariates on the treatment. Then there is the positivity assumption. Every patient needs to have the probability of receiving the treatment that you are studying, not having any, not being excluded from the treatment from some contraindication or something. And then there is, as I said, the consistency assumption, which in my case, as I have seen it now in practice, becomes a nightmare in a multi-country study. Like how clear the definition of your treatments are and your treatment strategy and so on. So what I'm trying to say is that if any of these assumptions by reality, by the real world, by the practice gets violated, your propensity score methods will be problematic and you will have biased results. But the statisticians, we still have hope in the sense that we can use diagnostics to detect whether, some of these assumptions might have been violated. And positivity, you can get some hints if you apply weighting propensity score methods by extreme weights, by some specific graphs. And there, that gives you alerts of perhaps trying to apply some trimming or truncating, but then you have to be careful what does that make to your Estimand? What are you left with? How many, patients you chopped out because they might have, done something to your positivity assumption?
I think positivity assumption sounds kind of easy, yeah. Well, but it's actually not. I've seen, for example, in an oncology study where certain patients had contraindications to treatments. Yeah,
exactly.
And of course, it doesn't really make a lot of sense to compare, to include these patients in your comparison because they'll never get the other treatment. Yeah,
exactly.
But it could also be what you mentioned in real life HTA things. Yeah, that maybe all patients first need to step through one treatment and then go to the other treatment. Yeah. And then, of course, nobody that hasn't gone through the first treatment can get the second treatment. Yeah. And so this kind of understanding in the real world is really, really important. So checking on the data is important, but also understanding what are the local guidelines, what are the typical kind of problems, what are the standards of care is really, really important. And you don't have that in clinical trials because there you just tell how to treat patients.
You design the perfect experiment and you mandate everything through your protocol, right?
Yeah. About the exchange, that is, if I understand it correctly, on the treatment side. Yeah, so basically that all the patients that you, in your database say get treatment X, all get the same treatment X, isn't it?
The extensibility, as I understand it, is more about the measured, the same treatment. I think it's about the consistency assumption. So one thing that often I see that we neglect is that, we applied propensity score methods. Yes, but on your measured, what about, this? It's a fundamental assumption. And this year, if I'm not mistaken, there was from Duke Markley some initiative together with the FDA on negative controls, on trying to leverage epidemiological tools in order to, check, do I have unmeasured thing? And to detect whether, I applied the propensity score, is my inference robust, and how sensitive it is to unmeasured confounding. And I found that fascinating. So these tools are there for decades, but I personally have never heard about negative controls. But I think there is an amazing opportunity on, the wealth of real world data to also investigate those negative controls in order to enhance all this inference about the propensity score methods. Because imagine you go to a health authority and you say, I applied propensity score methods, but on the top of that, I've also adopted negative controls to show you that, my methods are robust, or they are not. I think we owe that to be transparent and rigid in such evaluation when we try to infer causal treatment effects from such data.
Completely agree. And the estimate framework really nicely kind of lays it out. Yeah. That basically you have the same estimand then, but you do sensitivity analysis here.
Exactly.
Yeah? Because you check on your assumptions and you put in different assumptions. And then you can see how robust they are. How much kind of unmeasured confounding do you need to have to basically invalidate the results and come to something completely different from a conclusion point of view. And then you can think about, is that sensible or is that pretty unlikely to happen? Awesome, very good. If let's go through the ICH framework. Yeah, step by step. We have the treatment, we have the population, we have the variable, we have population level summary, and handling of intercurrent events. So we already talked that treatment there's surely difference between clinical trials and observational studies. Yeah. And that could be vary between the different countries, things like this. What else could happen in observational research that usually doesn't happen in clinical trials in terms of managing treatment in this Estimand framework?
I think also if I may say, the treatment history or treatment in a clinical trial, you highly select the population, exclude specific, perhaps pre-treatment history and stuff in the real world. No, although you can go in secondary data and try to apply the same eligibility, inclusion, exclusion criteria of trials. But then with treatment also, and that though I think it overlaps, and I will talk about this more when we go into the intercurrent events. Then you get, all the issues of adherence, compliance, switching treatment. But if you prefer, we can talk about that more when you ask me about the intercurrent.
And these kind of two things are closely related, especially in observational research. I think in clinical trials, you usually have provided in your protocol what to do, what are kind of the different steps in terms of changing treatment. But in real life, lots of different things can happen. Yeah. Yes. So just the variety of different treatment patterns, is much better, much bigger. And if you kind of think about that you want to do estimands based on these real treatment patterns, and then you are for sure an observational setting in clinical trials. So you really have this kind of problem with propensity scoring and all these kind of different things. So that's what I mean by observational. Of course, it's still a clinical trial, but then you have these problems, and clustering even kind of, different treatment patterns together is a topic. It's kind of, okay, for patients that don't know, let's say, switch from treatment A to treatment B within a certain time period, where maybe in a clinical trial, you only allow that at a certain time point, or if a certain criteria is met. All these different things are different in real life. And so it's just define what treatment is, when it's not just treatment policy, is really, really much more difficult in observational research. In terms of the population, you also mentioned kind of pre-treatment is surely one of the topics. It's kind of generally observational studies have much more heterogeneous patient population compared to clinical trials usually. Where else do you see problems in terms of differences between clinical trials?
I've seen a very nice paper specifically on this topic on Estimands, on trying to extend what we discussed on studies, keeping everything else steady, the current events, the treatment, all the 4 other attributes, just talking about the population. And it talks about that in the population element for observational studies, inherently you have also the covariates, because that's your fundamental thing in observational studies because of what we talked, the lack of randomization, this baseline covariates. You are trying to achieve the balance between the treatment arms. And that in observational arms, which differs from clinical trials, are these average quantities that they are not equals. The average treatment effect, the average treatment effect among the treated, the average treatment effect among the untreated. In observational studies, these are not the same quantities. In clinical trials, you wouldn't be worried about the inequality of this. Then different propensity score methods, they are they are coupled with this, like pair matching can only give you average treatment effect among the treatment. Full matching can give you both. And sometimes I've even seen when you apply caliper on the propensity scores, you lose that quantity of Estimand because you put that value to achieve the balance. And sometimes I've seen people going and comparing these methods, but you are not targeting the same. And also those Estimands are coupled with different research questions. So if a question concerns a treatment policy intended to apply to all qualifying patients, then your target population should be the whole indicated patient population, and the Estimand should be the average treatment effect. If the question concerns a policy of withholding a treatment among those currently receiving or not receiving it, the Estimand should be the average treatment effect among the treated or among the untreated. So, and then it's also what kind of data you have at hand. If you have a product, then you are confined only to the average treatment effect among the treated.
Yeah.
So those elements, I think they are, one can classify them under the population attribute of the ICH. And these are the fundamental differences to study.
Let's go to the third one, the variable, the endpoint that we look into. So the first thing that comes to my mind is that very often we don't even observe these highly specialized endpoints in observational research that we have in clinical trials. Because you need a lot of specific interventions, or you need educated physicians that can actually collect these data and these kind of things. Or you may have, a claims database where you don't collect these kind of things at all. So that's the first thing that comes to my mind. What other things do you see are difference between clinical trials and observational research with respect to that?
In endpoint, you mostly cover the choice of endpoint in observational research does not just depend only on the research question, but on the available real-world data sources. And within those, you need to understand how those measures or variables that they list, they were derived, through records of the physicians. Did they apply some machine learning? How did they extract this data? And for instance, former colleagues in Roche, I've seen yesterday, they have a very nice website and even R code on calculating real-world PFS, progression-free survival.
Oh, okay.
Because that can vary by cancer indication, by treatment, by you name it. And so they have a very nice case where they've studied such endpoint between a clinical trial and between real-world data, the Flatiron, which are amazing real-world data. And they found that for chemotherapy, the survival was pretty much the same, but for immunotherapy, it was not. And there are so many things to consider over there, what you were talking about before, the time window, what sensitivity analysis you do, and they have a very nice guidelines of how to go about with experts, with what is already known. So basically, they say that the mechanism of data collection differ between real-world data and clinical trials. And for instance, in the case of the PFS, progression-free survival, in clinical trials, tumor assessments are scheduled at regular intervals. Progression is assessed using standardized criteria. But then in routine healthcare, there is no protocol and no regularly scheduled tumor assessments, and progression may or may not be assessed following standard criteria. And this is why, for instance, we talk about real-world PFS and clinical trial PFS, and they recommend more studies of trying to acquire more empirical evidence per cancer indication, per treatment, to make sure that, let's say that you're interested in an external control of PFS. Are your endpoints similar? And then from from my previous experience, having worked a lot on measurement error issues and patient reported outcomes, there I can also see a lot of issues. Yes. Like, how did you collect did you record the quality of life? Did you use the same questionnaire? And even if you use the same questionnaire, did that same questionnaire have the same number of questions? Because in some questionnaires you have short or long versions of the scale. So if you look in different real-world data sources, how do you pull all these? And there are methods. One could look into harmonization of such measures. But in my experience, in 3 years in the industry, I haven't seen such techniques, to be honest, to be implemented or even discussed.
There's one thing that you just mentioned that I think I've never seen, more elaborated on is the variability. Yeah, the variability of the endpoint itself. Yeah,
yes.
That is can be much more harmonized and structured in clinical trials because you have training and all these kind of different things. But the variability, can be much bigger in observational studies. Yeah, lack of training, or because it's not exactly all at the same time point, or because the patient population is different. There's so many different factors that can influence that the inherent variability within your endpoint is very, very different, and therefore, of course, that has an impact on how likely it is that you'll see treatment differences and all these kind of different things. Yeah. Also, if you think about any kind of standardized measures, standardized by deviations, then of course that has an impact as well. By the way, probably the same is true for measurement errors in the covariates. That is yet another completely different topic.
Yes.
Which could be different in clinical trials to observational data. There's one thing that, however, I think is really good about observational data is that in observational data, of course, you have also these endpoints that people will see in clinical practice. Yeah. So at least if you have some kind of, medical data in it as well, not just the claims data, but you can see kind of patient-reported outcomes there. You can see endpoints like how long do they stay on treatment, if that is of interest in your indication. It could be, especially for good working treatment. If patients stay on it, it works. If they don't, it doesn't for them. So these kind of things can be amazingly nice for clinical trial, for observational research, and maybe much harder to actually study in clinical trials because these are studies. The treatment is provided. It's not, coming through the reimbursement criteria and these kind of things. There's usually no copay by patients and all these kind of other things that happen in real life. And so there are some areas where clinical trials, of course, are definitely better. But there's also some areas where observational research has its advantages with respect to variables. The next topic in terms of the ICH framework is population level summary. Do you see any differences there between clinical trials and observational research? Okay.
Yes, because yes, I do. In clinical trials, again, because of the randomization, the results bear statistical interpretation, and simpler statistical methods might often be required to estimate the estimands. But in real-world data, we need causal methods often to account for issues with non-randomization, and we can only interpret causally the results if the causal assumptions hold. So I think that per se creates what we were talking before. We have a duty to, verify the inherent assumptions of the causal methods that one might use. And then another thing, very, very simple, just mathematical fact, the collapsibility might complicate many important areas in clinical trials. When you have, a logistic regression model or a Cox regression model, odds ratios or hazard ratios, if you adjust for covariates in clinical trials, there are issues with the standard errors when different baseline covariate adjustment sets have been used. But if you do that, it's not only about the precision adjusting for covariates. It's also the problem of and that comes in terms of Estimand. Do I aim for a marginal Estimand or a conditional Estimand, right? Those are different things. With marginal Estimands, we would have everything that we talked before, AT, ATT, ATU, and so on. I'm not going to go over there. But if you are in the land of non-collapsible effects with odds ratios and hazard ratios, it's not the same. I run a Cox regression model and I adjust for covariates, or I run a weighted Cox regression model with as weight as the propensity scores, because the one is giving me conditional effects and the other is giving me effects.
That is really, really good. Yeah, you talk about odds ratios or hazard ratios in both cases, but really it's different.
Yes.
Yeah.
And I've seen many times, I've studied an adjusted Cox regression model and then comparing that with the clinical trial. You can't. Because normally a clinical trial gives you a marginal effect, let alone, what population, what the inclusion-exclusion criteria. Let's not go there. But, yeah.
This is brilliant. Yeah. And lastly, of course, we have the different intercurrent events. And we talked already about lots of different things there. What haven't we covered yet?
Yes, I think we haven't covered that in clinical trials you would have, and observational research, it would be the same for intercurrent events, the lack of efficacy or the safety. But then in the observational research are the personal factors, the relationship that the patient has with his physician, or a family member, influenced me to get this treatment that you probably wouldn't have in a clinical trial, or at least in the clinical trial, perhaps in the protocol you would have recorded it. And then also events related to non-behavioral factors like change of health insurance program, relocation to another place where the current therapy is unavailable, development of new conditions that contradict the use of the current therapy, participation in a clinical trial that requires discontinuation of the current therapy.
A lot of additional things to have in mind when you write your intercurrent event strategy. Yeah. Awesome. Thanks so much for going through all these kind of different things. And it is super helpful to understand what we can learn about the Estimand framework from observational studies, from clinical trials, and how they actually get together. And it was really, really interesting to understand kind of how treatment, population variable, population level summary, and intercurrent events all differ for clinical trials to observational data. Where do you see the future of real-world data research going as a result of this Estimand framework?
I think, as we discussed, with real-world data, we have to take into account many additional considerations in relation to Estimands. We also have to understand the stakeholders and their question of interest, and then accordingly evaluate how fit for purpose the real world data that we have at hand, or we can find, or we can seek, are, coupled with all these issues. And definitely articulating each of these 5 attributes can help identify and define appropriate Estimands. And we should definitely not miss other frameworks that they're closely related, like the causal inference or the target trial framework. And basically to align the analytical methods with selected real-world evidence study estimates, and definitely apply rigorous sensitivity analysis to ensure the robustness of the study findings.
Thanks so much. That was an awesome outlook and summary for this podcast episode. Thanks for being on the show, Artemis.
Thank you so much. Thank you, Alexander. It was a pleasure and honor to be here and discuss these things with you.
If you want to learn more about Estimands, then check out the Effective Statistician Estimand course. You will find a link to this in the show note, and you can also look for it on the effectivestatistician.com homepage. This show was created in association with PSI. Thanks to Rain and the team at VVS who helped with the show in the background, and thank you for listening. Reach your potential, lead great science, and serve patients. Just be an effective statistician.